Hi, I'm Sonika Tyagi, presenting on behalf of my team at the School of Computing Technologies, RMIT University, Australia. I'm going to discuss integrative machine learning frameworks for clinical outcomes from multimodal data. In order to enable precision medicines, the holistic understanding of a biological or clinical system is required. And this would involve two components. One is to combine multiple layers of a genomic systems where you could combine blocks of various biomolecules, for example, gene, protein, metabolites, and so on. And while we are doing the integration, we make sure to take into account the intra-omics correlations as well as between omics correlations. So what we are doing is simultaneous integration as well as correlation of various information between different omics blocks. So what this allows us to do is to first capture the lowest level interactions between different entities of a biological system, as well as uncover new knowledge that was not possible by analyzing these individual blocks of omics separately. You could do such analysis of various biological systems, for example, protein interactions, drug interaction networks, drug and disease interaction, non coding RNA, and so on. So, by building these various multi-omics blocks, we get a system level understanding of a biological system. Now, how about we can overlay other related life and health related information of an individual to build the complete system in order to study diseases for precision medicine and treatment. With you today, we have developed methods that allow you to integrate various types of functional omics blocks, which is mainly numerical measurement of, let's say, gene in the form of transcripts, proteins, metabolites, lipids. So any omics measurement that you can represent as a block of numerical table. We also use natural language processing to process information at the genomic level, which is another omics layer. And we work on a hypothesis that regulatory signature made of these various sequence level signatures derive the functional omics signature, which is made of these expression profiles of various biomolecules. And by combining various blocks, we can get a 3D understanding of a cellular function. On top of that, we plan to uh, integrate data from health domain. So these are the electronic medical records of an individual that are stored through healthcare facilities. And here our effort is to build systems that are reproducible, scalable, as well as generalizable. So there is a lot of emphasis on building machine learning models that can work in unseen situation with a robust performance. So here I'm presenting a workflow called EHRML, where we allow various modules to do an optimum design of a machine learning modeling technique, allow you to select dynamic window of data observation, as well as you can play with various parameters and hyperparameters of your study design. And at the core of this integrative machine learning framework is the ensemble architecture that we allow, where we allow a flexible design where a user can select the machine learning models of their choice, and we allow multiple fusion architecture to combine the predictions from different models. So here I'm showing an ensemble architecture where we do a two-level integration. In the first level integration, we allow prediction from multiple individual models, and in the second layer, we use those predicted probabilities as an input to do feature representation learning in the second stage to do final predictions of clinical outcomes. And we have shown this is an outcome for mortality prediction of a patient cohort. And what we are showing in on the left side plot is that the ensemble model outperforms, so which is the yellow line here, it outperforms all of the individual standalone models. And it also works very well in the situation where we have very heavy class imbalance, which is normally is the case when you are where your predictor of interest is always in a rare amount. And it works very well in under various conditions. And behind all these integrative framework is also a standardization and data pre-processing pipeline that works behind the scene. So there is a pipeline called EHRQC that allows you to streamline automated pre-processing of health records, and that works at two levels. One is the standardization level and the pre-processing. So in the standardization module, what we are doing is to standardize the format of the data itself, because every healthcare institution may store data in their individual institution specific format, as well as harmonizing the medical concepts themselves. These are the terminologies that are used to represent diagnosis, treatment, medications, and so on. So we apply control vocabulary to perform concept mapping. So in this case, we do two-level standardization, and then standardized data goes through the pre-processing pipeline that does a comprehensive quality control analysis, as well as allow you with various to do data-driven pre-processing to remove, let's say, missingness, outlier, or any anomalies that are observed, and it gives you a comprehensive report over there. And while we are doing this data standardization, so on the left-hand side in this block is the source data from a healthcare facility, and on the right-hand side is the standardized data. So while we are performing this standardization, we make sure that the entire pathway to data transformation is completely transparent. So if there is any data loss, that's completely interpretable and explainable. On the right-hand side, what we are showing is the automated concept mapping using controlled vocabulary databases. Again, it's a flexible system. We use natural language processing and combination of multi-voting approach to automatically map the control vocabulary concepts to the known concept in a healthcare data system. And we have shown that this our methodology outperforms expert curation, which is currently the standard in this space. So therefore, we have shown that that there is requirement to combine data from multiple sources, or both at the biological level as well as medical or health level. And this data is now routinely available in large cohort studies, and allows us to uncover new knowledge by matching information across multiple blocks of data or multimodal data. But in order to enable such kind of integration and uncover knowledge for precision medicine, we need to give a lot of emphasis on adopting standards as well as building pipelines that are scalable and reproducible, so that we can build these intelligent machines that can perform very well in clinical settings and can be generalized to wider population of that nature. Thank you for your attention.